today, about 13 questions from that chapter. But it's going to be done as basically a self-study chapter where I want you guys, I don't give much homework, okay, as you know, but I want you guys to review that information on your own. I will include that material in the review. Like when I give a review, I will tell you what to kind of pay close attention to on that chapter. You remember how I did on the test that one chapter where we didn't go over it, but I said, look, you need to pay real close attention to nephrotic syndrome. There's going to be a question on the test about nephrotic syndrome. Remember, I covered all those, yeah.
And so, in this case, chapter 8, urine screening for metabolic disorders.
Yes.
Chapter 8, urine screening for metabolic
There's a
disorders.
lot of information in that chapter, and I wish we had time that I could go it thoroughly with you, but we're behind a week. Okay, for whatever reason, I don't know, but we should be one chapter ahead, and so I'm catching up today. Okay, I'll catch that up. So we're gonna cover spinal fluids, because in my eyes, spinal fluid is a lot more important than you being able to diagnose somebody with least knee hand disease or equivalent disorder. Okay, because that will not really be what you do when you go to clinicals. We will talk about, Bianca, I'm sorry, I put all that stuff there, I was thinking that table. So anyway, that's gonna be kind of a self-study chapter. Do not ignore it. Make sure you read over it, and again, I'll include it in the review. Our next test in here, even though you took a test last week, our next test will not be next Friday, but the next Friday. We'll have our next testing, all right? It will end with semen analysis. The chapter on semen analysis, which we will cover next Friday. And then after we get done with semen analysis, we're gonna go into the lab. I'm gonna try to put something together where you guys can simulate testing spinal fluid, all right? And then after that, we'll continue on with our papers. I'm planning on having about 30 extra minutes today for you guys, for any of you that are possibly ready to present your PowerPoint.
Yes.
Is there anybody, I'm not saying we're gonna get to everybody, but who all in here is ready today to do a PowerPoint if we have time? Okay, that'll be toward the very end of class, all right?
So I can go through mine real quick.
Mr. Turner.
Yes. That's what we're coming up with, not next Friday, but the next one. Next Friday, I think next Friday is November the 1st, and so, yeah, November the 8th sounds right. All right, before we get into today's chapter on spinal fluid, I just want to give you a little bit of a broad interview, just talking with somebody that's tested a lot of spinal fluid before. Spinal fluid is kind of like a sacred substance in the lab. It's not real easy to obtain, so it's not one of those specimens that you want to contaminate. It's not a kind of specimen that you want to drop, not a specimen that you want to lose, okay? Because it takes a lot to get a specimen that has spinal fluid. Have any of you ever known someone that's had to go to the hospital and have what's called a lumbar puncture? That's where they go in and they obtain spinal fluid, all right? They go, I think I've seen it done once, but they go in and basically tell someone to kind of get in a cradle position on a tabletop, and they go with a needle kind of in your back, okay? And then they go and they obtain spinal fluid. They can't always get as much as they want to, even though the book is going to present itself in a way, and I'm going to go over it where it says this is what you do in tube 1, this is what you do in tube 2, this is what you do in tube 3. But a lot of times you'll get one tube sent to the laboratory and it'll have just a little bit of spinal fluid, and that's all they could get because maybe the patient had a real high intracranial pressure when they were doing the procedure, all right? So it's not always going to be one of those things to where you have a lot of fluid to work with.
My son had to have it done twice. They thought he had meningitis.
Yep, that's one of the main reasons they do spinal fluid is they are checking for some sort of meningitis. There's bacterial meningitis, there's viral meningitis. I'm sure in Jason's class, you guys have studied Neisseria meningitidis. You guys ever went over anything like that?
Yes, sir.
Okay, another thing that the book really doesn't present, but I want you guys just to kind of hopefully take away from today's class and possibly next week in lab, is that because this stuff, you don't ever throw it away, first of all. Like when you get finished using it, I don't care what department you're working in, if they got a gallon of spinal fluid, okay? This is not something that you throw away in a week, because a lot of times they may wanna come back to that. I can tell you right now, if you go to CHI Hot Springs, they have all of the spinal fluids that have been tested for a year. They keep them for 1 year. It doesn't matter what department, when you're finished with the specimen and you think you're done with it, a lot of the departments, when they're completely finished, they will freeze it and put it in a freezer, and it is kept there for a long, long time. Okay? Like if I wanted to go get a spinal fluid, I probably could from them. But another bad thing about spinal fluid is after a couple of hours, the cells start to either disintegrate or they go away. So you can take a spinal fluid that was loaded with white blood cells, say 3 hours ago, but if you wait a real long time in testing it, some of those white blood cells and red blood cells may have lysed. So now they're not there. And so pretty much all testing that is done on spinal fluid is performed stat. The only test that's not performed stat on spinal fluid usually is gonna be in microbiology where they order cultures on spinal fluid. Of course, it takes cultures 24 hours to grow, so that's not stat. But if you're like I was, say you work at St. Joe in the one department that you don't work in is microbiology, if you work an off shift, like swing shift or graveyard shift, if they happen to do a spinal fluid on a newborn coming through the ER, it's one of the few times that they will order a stat gram stain. So you'll have to know enough, at least about micro to go in micro and do a stat gram stain. They want those results very, very quick because like Wendy just mentioned, what they're checking for a lot of times is meningitis on that baby. They want those results fast. Another thing that's real important about that is a lot of times there won't be a lot of organisms in that spinal fluid. And so you have got to be really, really good at focusing the microscope. It's one of those things that you can't miss. You do that gram stain, you can't just be looking in there and go, nope, it's negative. It's like, even if there's just a few of those Neisseria bacteria or strep bacteria, you've got to see them, okay? You've got to say, look, there weren't many, but I'm seeing organisms in the spinal fluid.
Isn't that they should be sterile, right?
Right, spinal fluid for the most part is like blood, it's sterile. Yeah.
That's one of those situations where you want to check 10 fields.
Definitely. And you also want to, like, if you're having trouble with that microscope, you don't want to just go, oh, it's probably negative. You want to make sure you're seeing it. You can get in trouble if they find out that baby had some sort of bacterial meningitis and you didn't catch it on the gram stain. Okay. That's one of those situations where they'll go back and look at your slide like a supervisor to make sure that they weren't there. Sometimes a child can have meningitis and there are no organisms or bacteria on the gram stain, so you didn't do anything wrong. But if they go back and look at that slide, you just didn't have it on focus or you didn't catch it or something like that, you can get in a little hot water for that, okay? So just letting you know, spinal fluid is one of those things where you need to be very, very careful with it. Also, it's going to tell you in your book, because spinal fluid can have meningitis in it, you want to be very, very careful when you're handling it. You always want to have on gloves when you're dealing with spinal fluid specimens. Okay, so starting on page 182, the very first 4 lines is the answer of your first objective for spinal fluid. This answers what are the basic functions of spinal fluid. On page 182 at the top, it says CSF is a major fluid of the body. CSF provides a physiologic system to supply nutrients to the nervous tissue. It removes metabolic waste, and it produces a mechanical barrier to cushion the brain and spinal cord against trauma. One of the things they worry about a lot of times in spinal fluid from car wrecks and things like that is there a bleed in the brain, right? Like did they hit their head hard enough that they've got bleeding that's going on within the brain, and you can find that out if you see a steady amount of blood in the spinal fluid. Okay, the formation and physiology. I want to talk about this a little bit. There's a great picture at the top of page 183 that shows the brain and the spinal fluid and all the lining membranes, the dura mater, the pia mater, and it explains this under the formation and physiology. The first sentence under that says the brain and spinal cord are lined by something called meninges, okay? Now that should help you to remember, think about it, bacterial meningitis, inflammation of the meninges. Something is going on in that bacteria. Think of it as it's causing an inflammatory response, and that inflammation is causing the meninges to be infiltrated in a roundabout way, and that can cause damage to what's called the blood-brain barrier, which we're fixing to talk about that as well. Okay, next paragraph. CSF is produced in the choroid plexuses of the two lumbar ventricles and the third and fourth ventricle. It says in adults, approximately 20 mils of fluid is produced every hour. The fluid flows through something called the subarachnoid space. All right, the very last sentence in the left-hand column. Go down to the very bottom line. It says in the choroid plexuses, there's something called endothelial cells that have very tight-fitting junctures that prevent the passage of many types of molecules. This tight-fitting structure of endothelial cells is in the choroid plexus is termed the blood-brain barrier. It says maintaining the integrity of the blood-brain barrier is essential to protect the brain from chemicals and other substances circulating in the blood that can harm the brain tissue. If you'll skip a sentence, it says that disruption of this barrier by diseases such as meningitis, multiple sclerosis, allows white blood cells, proteins, and additional chemicals to enter the CSF. I'll tell you a story about this that can kind of maybe help you to remember it. I had a doctor one time prescribe me a drug called Inderol. It was one of those deals about my heart. I was having some issues with it. And anyway, so I started taking this drug called Inderol, the heart medication. And I took one pill of it, and I was out in the country driving around in my truck or something. And all of a sudden, I just started feeling like weird. You know what I mean? Like I could tell, like, whoa, I need to pull over. I just felt so weird, and I felt like just, like I didn't have the energy to do anything. Like I remember I just pulled over and like laid my head down on the side of the door because I knew I wasn't like having a heart issue, but I just felt like I didn't have the energy to do anything. And so I went back to that doctor because I'm like, I'm not taking that drug anymore because whatever is in it, it like just made me completely just useless. I didn't want to do anything. I just felt like I just wanted to lay there and never get up. And then later on, he had told me that drug, they had done some testing on that drug and they had found out that it can cross the blood-brain barrier. And so it kind of gets in your spinal fluid, and that's why I was having all those side effects. Needless to say, I wasn't ever going to take that drug again, period, no matter what. But just saying, that's what they're talking about when they say your blood-brain barrier a lot of times protects your spinal fluid and your brain from some of the chemicals that we might take, okay? Like whatever drug you're being prescribed, there's a real good chance it's not supposed to cross the blood-brain barrier, okay? Or else you may have a lot of side effects to that. If that kind of helps you understand how important it is. And of course, again, any blunt trauma to the head can break that blood-brain barrier. And that's where you don't want bleeding into the brain, where all that pressure builds up at. Okay, specimen handling and collection. I've already looked at the test, and there's about 3 test questions just off this next couple of paragraphs in the book. The first thing is it says CSF is routinely collected by a process called a lumbar puncture. So make sure that you have that highlighted. And it goes on to talk about the pressures and things like that. But one thing that they're real strict on, one thing that is of high importance in the MLT program when it comes to spinal fluid, for whatever reason, is they want to know if they put spinal fluid in tubes, they number the tubes, and what they want you to know is which department in the lab gets which tube, okay? So what I mean by that is usually if everything is going good and they do a lumbar puncture on someone, there's going to be tubes that are numbered 1 through 4, okay? And for the textbook, and most textbooks are going to tell you, for example, tube number 1 goes to the chemistry department. Tube number 2 goes to microbiology. Tube number 3 goes to hematology. And if there is a tube number 4, a lot of times they want that tube to also go to microbiology. Okay? Now it tells you this in your book. It specifically states all this information. I'm on page 182, right-hand column, go down to the very bottom. You're you're gonna see a bullet point that says tube number 1 is used for chemical and serology tests, because these tests are least affected by blood or bacteria introduced as a result of the tap itself. When they say the tap, they mean the actual procedure. Everybody with me on that? Tube number 2 is usually designated for micro. Tube 3 is used for the cell count, okay, and that means hematology. And then it goes on to say a 4th tube may be collected for microbiology. Now, if you go on down to the next set of bullet points, it tells you that if you cannot perform these procedures stat, if you can't do them stat, they tell you how they want the specimen preserved in each department. See how it says hematology tubes are refrigerated? And it says microtubes are left at room temperature. And then chemistry and serology tubes are frozen. The picture on page 183 at the bottom, that's a good picture to study by. Like if you look at my book, see that tube 1, I've got written right beside it, frozen. Tube number 2, micro, I've got written right on that tube. That tube would be kept at room temp. Tube number 3, refrigerated. All right, another thing that I want you to be aware of, and it will not state this in this book, but I've seen it happen too many times. A lot of these physicians that do spinal fluid analysis and have to perform lumbar punctures, sometimes you will get what they call in the lab a downtime, and they will handwrite what they want done on each tube. Okay, so even though I want you to study this book, and when you take your registry and they say, look, tube number 2 for spinal fluid should be sent to which department, I want you to remember this. But I also want you to know when you go to clinicals, there are some doctors that are going to handwrite on a piece of paper, and if they are going to tell you what tests they want done on each tube. In other words, they may not agree with this format. Okay, I'll give you a great example. And it tells you this in the book in a roundabout way. A lot of doctors, they say, look, I want a cell count and a differential done on tube 2 and tube 4. And what they're doing is they're trying to figure out, is there more blood in tube 2 than there is 4? Because if there is, this may be what they call a bloody tap. That means they got blood into the specimen trying to get down to the spinal fluid. Does that make sense? And I'm fixing to go over some little things you could possibly look at to determine if you have a bloody tap. A bloody tap basically means in tube 1 and 2, you're gonna have some blood in there that truly are not in the spinal fluid. It's they got blood in the specimen because they had to stick that needle through little small vessels and stuff trying to get into the spinal fluid, all right? So, I mean, it'll make sense, I think, once we go through it. Now, another thing, remember, and I think that there is a note on this, turn to page 184, because I think this is real important. It's under the, on the very top of page 184, under the technical tip, it says, if only one tube can be collected, it must be tested first by microbiology. You remember I've told you guys this in other lectures before, if you are ever dealing with a fluid that's hard to collect, whether it's spinal fluid, whether it's amniotic fluid, whether it's pleural fluid, synovial fluid, if you only have a small amount of fluid and you only have one tube, always go to micro first with it. And even if they say they don't want it, like, what are you doing back here? We don't want that specimen, okay? You give them the opportunity and you say, look, I've only got one tube of this spinal fluid. Do you guys need it? Because if you take that tube to chemistry and they're doing like a total protein and a glucose on that spinal fluid, guess what happens when they're putting a squeegee pipette in there and transferring it? You just contaminated it, okay? And so always, if you have a small amount of a fluid, always take that to micro first and ask them if they need it. Because if they contaminate it, right, and then they do a culture on it, do you understand that they could grow out normal flora? Or they could grow out something simply because they contaminated that specimen. So that's real important, that technical tip up there. And then the next box on the technical tip kind of refers to what I just told you. It's not unusual for cell count requested to be performed on both tubes 1 and 4 to check for cellular contamination by the puncture. All right, another answer to one of your objectives in the first of the book, it says, what should the appearance of normal spinal fluid look like? There. I'm on page 183, left-hand column, go down to appearance. The very first sentence says, the initial appearance of normal, crystal clear CSF can provide valuable diagnostic information. So, I do not have any real spinal fluid in any of these, but since we're not doing lab today, I want you guys to get a visual of this, because this is what spinal fluid tubes look like down in the lab, okay? They'll be just like this. Everybody see that? Now, I've got one, because a lot of times you're not gonna have a lot of fluid. Can you guys see? I mean, I just put water in it, but do you see the crystal clear fluid in that? That's what spinal fluid should look like, and that is about how much usually will be in each tube, right? So what I would do, because you guys may not be able to see it from there, but if you turn this tube around or peel back the label, you guys can't see it, but there is a circle right there that clearly says this is tube number 4, okay? Hence, you have to look at these tube numbers and take them into the departments, tube 1, 2, 3, and 4. In other words, there's 4 of these that come in the kit when the doctor performs the procedure and takes the spinal fluid off. All right, so this would be tube 4. This one should not have a lot of blood in it and stuff unless there truly was blood in the spinal fluid, okay? Like if I look at this other tube, I'm gonna talk about it in a minute. Well, as usual, this is a tube 4 also.
Question.
Yeah.
Those two tubes would go, if requested, to the microbiologist.
If they didn't tell you where to take them, tube 4 is kind of like an additional tube. They're really worried about the first 3, but if they did get a good tap and they had enough to get a tube 4, this would go, I think it says in your book, this would be an additional tube that could be sent to microbiology. All right.
They come in a kit of 4.
What's that?
They come in a kit of 4.
Well, I believe so. I mean, to be honest, I've never seen them actually pull spinal fluid. I've been on a lot of bone marrows, but I haven't seen the spinal fluids.
Okay.
But yeah, those come in a kit. They don't keep them in the lab. That's kept on the floor at the nursing stations.
Okay.
Okay, page 184. I'm just underneath that picture on page 184, figure 9, tab 4. You've got a bolded word there called xanthochromia. You're gonna need to always remember this word, okay, xanthochromia. This is a term used to describe spinal fluid supernatant. What is supernatant?
After it's been spun down.
After it's been spun down, okay? If you take a spinal fluid and spin it down, if that spinal fluid is pink, orange, or yellow, if you hear a tech describing the spinal fluid that's supernatant as that color, they're not gonna say it's yellow. They're gonna say it's xanthochromic, okay? So go ahead and bite the bullet and learn that word, because you will see it again, okay, a couple different times. It says a variety of factors can cause xanthochromia, the most common being the presence of red blood cell degradation products. Now, why is that important? Red blood cell degradation products, that's just a complicated way of saying the red blood cells have broken down. They've been in the fluid long enough that they're starting to degrade. That's real important, because if we see a spinal fluid that's that color, that means that blood has been in that spinal fluid for more than 2 hours. Okay, so why is that important? Because if that physician gets blood into the specimen because it was a bloody tap, you see what I mean? Like they just had trouble getting the specimen, those red blood cells have not been in there long enough to cause the specimen to be xanthochromic. But if that person has had a cerebral hemorrhage, maybe they had a cerebral hemorrhage 4 days ago, okay? So the spinal fluid truly has had blood introduced in it, then xanthochromia can tell that physician a lot of information, okay? Now again, The only true way you can tell if a spinal fluid specimen is xanthochromic is you have to spin it down and look at the color of the supernatant, all right? Now, let's just pretend that we spun this down. This, I tried to make this as perfect as possible, but this would be called a xanthochromic specimen. See that yellow color? Remember, normal spinal fluid should be crystal clear. This would be an excellent example of a xanthochromic specimen. Yep.
And so, aside from supernatant, it also has its own little version of sediment in it too, correct?
Yeah, if you spin it down. But some departments, they don't spin it down. Like, you surely wouldn't spin down a spinal fluid to do a cell count on it, right? Because what would happen if you accidentally spun down a spinal fluid and tried to do a cell count? Where's all your cells?
The bottom.
The very bottom, right? So you want to make sure if you're doing a cell count, you want to actually mix it up, okay? And that's going to lead me to something else before I forget to tell you. If you're ever dealing with a spinal fluid, even if it's this much, don't do what I did one time, and I never made that mistake again, but I did do it once. There are certain procedures where we actually have to add dyes and stuff to the spinal fluid to do a cell count on it and things. If you're ever testing it, if you're working in hematology and you have this fluid and you want to test it, do yourself a huge favor and take out half of it and put it like in a blood bank test tube and test that fluid.
Okay.
Don't just all of a sudden start testing this, because if you contaminate this, you contaminated the whole specimen, right? So always just like take out half if you can and put it in a separate tube and let that be what you work with. Because if you mess up or you make a simple mistake on it, you didn't contaminate the entire thing, okay? That is really, really good information. Just trust me on that. And the reason I say that is because there are certain cell counts you do that you have to dilute it with a dye. And if you try to do it and do it just perfect and get all that dye in that fluid, you've just contaminated the whole specimen. We're going to talk more about that in a couple of minutes. Okay, on down in that paragraph where we were just talking about, it does say there are other things that can cause xanthochromia besides just red blood cell degradation products. They mention things like elevated bilirubin levels, the presence of certain pigments like keratin, increased protein, any type of a melanoma pigment, okay? But the thing that they worry about the most is the red blood cell degradation products, because they're worried about a cerebral hemorrhage. Okay, so let's go on and talk about a traumatic tap versus a cerebral hemorrhage, because that's an objective that you have also. I'm under traumatic tap on page 184 in the left-hand column. Grossly bloody CSF can be an indication of an intracranial hemorrhage, but it may also be due to the puncture of a blood vessel during the procedure itself. So one thing you can do is you can check for uneven blood distribution. It says blood from a cerebral hemorrhage will be evenly distributed throughout the 3 spinal fluid specimens, whereas a traumatic tap will leave the heaviest concentration of blood in tube 1 and gradually diminishing amounts to 2 and 3. So what I've done is I've shown you a perfect example of something that's called, this would be a classic bloody tap. I want you to imagine all these are spinal fluids, okay? Now I know I showed you the spinal fluid that they actually collected in, but let's just say you poured it off, or if they did it in a different place, so I'll put it here. This would be tube 1. Everybody seeing it? See how bloody it is? But see how in tube 2 it's starting to go away? See how in tube 3, very little, if anything, and in tube 4 it goes completely clear. This is a perfect example of what's called a bloody tap. This is not representative of the spinal fluid, okay? This is blood was introduced trying to do the procedure.
Right.
If it was truly a cerebral hemorrhage, it would look like, say, this in every single tube. It would have this exact color. Does that make sense?
Mm-hmm.
Okay. So if you have a lab practical for your final, there's a good chance that I'm going to line up some spinal fluids and I'm going to give you either an example of a cerebral hemorrhage or a bloody tap, and you should be able to recognize it by looking at it. Okay? Everybody good on that? Understand that? Yep.
Once again, what you got in your hand is a bloody tap.
Yeah, this is an example of a bloody tap. And a lot of doctors, and you will hate it working in hematology, because when you have to do manual cell counts on these, just trust me when I tell you it takes a long time. You gotta do them on the hemocytometer and all of that. They're gonna say, I want you to do a cell count on tube 1 and tube 3. And that takes a long time, okay? Because you also have to do a manual differential on that one and that one. Okay, under clot formation, I'm on page 184 in the right-hand column. Clot formation, it says fluid collected from a traumatic tap may form clots owing to the introduction of fibrinogen into the specimen. So that's another way that you can determine if it's a traumatic tap or if it's a true intracranial hemorrhage. Okay, some more about the xanthochromic supernatant on page 185, left-hand column, xanthochromic supernatant. It says red blood cells must usually remain in the spinal fluid for approximately 2 hours before noticeable hemolysis starts to occur. Therefore, a xanthochromic supernatant would be the result of blood that has been present longer than was caused by the tap itself. Skip down about 4 or 5 lines, and it says, to examine a bloody fluid for the presence of xanthochromia, the fluid should be centrifused in a microhematocrit tube and the specimen examined against a white background. Okay? So again, let's say we have this tube right here. Let's say that every tube looks just like this. But we don't know, you know how, like, for example, if you take a urine specimen and it's loaded with blood, if you spin it in a centrifuge, sometimes all the blood goes down to the bottom, all the red blood cells, but now the urine looks perfect, except for that big red button at the bottom, okay? A truly xanthochromic specimen, if we were to take a hematocrit tube, let's say this is our specimen, so we get a hematocrit tube, you guys remember what these look like. We put it down there and let some of the fluid go in by capillary action, and we spin it in a centrifuge. Now remember, we're just looking at the supernatant. If we have a bunch of red blood cells at the bottom, but now our spinal fluid looks crystal clear, okay, that is not a xanthochromic specimen, okay? But if the supernatant looks that color, now it is a xanthochromic specimen. You understand what I'm saying? So you have to spin it down. Make sure you understand that in order to make that call, whether it's xanthochromic or not. Okay, cell counts. 185, left-hand column, halfway down. It says the cell count that is routinely performed on spinal fluid specimens is the white blood cell count. They're not usually as worried about the red blood cell count as they are the white blood cell count. Okay? Now they may ask for a red blood cell count, but sometimes they're not even worried about that. They just want to know the white blood cell. Okay, skip down toward the end of that paragraph under cell count. It says any cell count should be performed immediately because white blood cells and red blood cells begin to lyse within 1 hour. 40% of the leukocytes disintegrate after 2 hours. Specimens that cannot be analyzed immediately should be refrigerated. Okay, in other words, we're talking about the hematology department. So this would be tube 3. Remember CMH, chemistry, micro, that's how they're distributed. Under methodology, on page 185, left-hand column, last paragraph, normal adult spinal fluid only contains 0 to 5 white blood cells per microliter. So calculating CSF counts. There will be a question on your test where you have to calculate a CSF count. So let's talk about it. Now this is going to be one spot that I don't really refer to your book because I'm going to tell you, in my opinion, the proper way to do this. Okay, but before we go over the counts, I do want to read you one thing. If you'll turn on page 186, left-hand column, purple bolded word, white blood cell count. Everybody see where I'm at? It says lysis of red blood cells must be obtained before performing a white blood cell count on either diluted or undiluted specimens. Specimens requiring dilution can be diluted in the manner described previously, substituting 3% glacial acetic acid to lyse the red blood cells, including a substance called methylene blue to the diluting fluid stains. It stains the white blood cell nuclei. Okay? So, they make a substance called this right here. This is very old, actually it's from 1994, so, clearly says spinal diluting fluid. This is diluting fluid for spinal fluid, okay? And just trust me, you'll see it next week. This is a real dark blue stain in here, and you don't want to get it on you, okay? But the good thing about it is there's two things in here. One of it is there's a real dilute acetic acid, 3% acetic acid. Do you guys remember what acetic acid does to red blood cells? Lysis them, okay? And there's also a substance in here, crystal violet, that if there are white blood cells in the fluid, the nucleus in the white blood cell is going to show up a little better than if it was just spinal fluid with nothing added. So you got two things in here that really helps with the fluid. So before we do this count, I'm just gonna, and we'll do this next week in lab, but I just want to at least kind of go over it where you have a basic understanding of it. Because we have technology now, a lot of times if a spinal fluid has a lot of color to it, if it's bloody or if it has enough cells in it, we can run it through the Coulter or the CBC analyzer and get a count that way. Where you don't have to count it manually on a hemocytometer, okay? The ones you have to worry about anymore are the spinal fluids that are clear. Because even though the spinal fluid is clear, that physician, he or she, is still going to tell you they want a cell count and a differential done on that fluid, okay? So you're probably not dealing with a lot of cells, all right? So because of that, most of the time, if you're having to count a spinal fluid manually, you're doing it on an undiluted specimen. So the best way to do that, before we start talking about the counts, is I would take this fluid right here. Let's just say this is my crystal clear fluid, all right? And let's say that they are wanting a white blood cell count and a differential done on this. First of all, I can tell you, just looking at it, it's not cloudy or anything, so there's probably very few cells in here. So what I would do when I did the count, I would do it straight. I wouldn't dilute it with anything, and I would pull out one of these little capillary tubes. I don't know if you guys can see this or not. Okay, this is a little capillary tube. It's got a blue line on it, and that blue line represents that there's no heparin in it, okay? It's a plain capillary tube. So what I would do is I would stick this down in the diluting fluid and let some of that stain get in here, and I would mix it like this, and you'll see the stain run up and down, right? And then I'd put my finger on it, and I would put it on a chem wipe and let all of that stain drain out of it. Okay, so you're thinking like, why did you do it then, right? What happens is that stain, there's not very much of it, but it will just be barely coating the inside of this. You see what I mean? Mm-hmm. Like I drained all the stain out, but there's just enough stain that's coating the inside of this that now I'm going to take this same capillary tube and I'm going to go down in my spinal fluid, okay? And I'm going to get just enough spinal fluid in there, probably fill it halfway up, and I'm going to mix it back and forth. And as you start looking at it, that spinal fluid will turn a light blue color, okay? Because now it doesn't have very much at all, not even enough to do the calculation, but that little bit of stain that was coating the inner layer of this is going to be mixed in with your spinal fluid. There's going to be enough that it will lyse all your red blood cells, and it will stain your white blood cell nuclei a purple blue color, all right? So that's pretty good, right? And now you hold your finger over it, and now this is what you load your hemocytometer with. Now, I think I showed you guys this trick in the summer. Remember when we were doing red counts and you made the dilution? And I said you can let some of that fluid wick up the capillary tube, and you can actually, remember the little V's on the hemocytometer? You can like let a drop get out and then touch the hemocytometer and fill up the chamber, okay? So this would be an undiluted specimen because we didn't put enough stain in there to cause it to have technically a dilution, all right? Alright, so let's talk about a white blood cell count. So let's just say that we did all this, and I'm gonna show you this next week. I'm not one of these people that like to just talk. We're gonna do it next week, and I'm gonna show you how to do it. We'll at least practice doing it. But now let's say we've got the hemocytometer loaded. There's a picture in your book that shows you, either turn back one or over, show you the grid. Go ahead. Yeah, right there. What page is that? I can't see. Yeah, turn to page 185. All right, so here's our hemocytometer. Here's one side, and then here's the other side. All right, now I'm gonna make the white blood cell count real easy for you, because what we're gonna do is on each side of the hemocytometer, you remember when over in the summer, Jason and I was telling you how we did manual counts on a hemocytometer to try to make the actual figuring out the result real easy? If you don't count 10 squares, okay, it makes it hard, because you've got to figure out what volume correction factor to add in on it. Okay, so I'm gonna make it easy for you. We're gonna count 10 squares, okay? There's not 10 squares on one side of a hemocytometer, right? So you can't really take both sides and take the average. But if you count one side and count these squares, how many squares did I count? 5. Okay. You go to the other side and you count these squares. How many squares have I counted? That's 10, right? 10 large squares is equivalent to a microliter, okay? The white blood cell count is done per microliter. So if you don't dilute it, if you do not do a dilution and you count 10 squares, however many white cells you counted, that's your answer, okay? So if I counted all 10 of these squares and I ended up counting those and I ended up with 27, 27 is my result. 27 white blood cells per microliter of spinal fluid. That would be my answer. Questions on that? That's real important, because I'm telling you, on a registry, they're gonna ask you one of these. Now, it may not be white blood cells in spinal fluid, but it's gonna be white blood cells in some type of fluid. Pleural fluid, pericardial fluid, something, okay? But you've got to understand a hemocytometer, the whole goal is to count 10 large squares. Where it gets confusing is if they tell you, okay, you only counted 4 squares on one side. Now you've got to do a volume correction factor. But all you're really doing, and now listen to what I'm fixing to tell you, this is real important. If they told you you only counted 4 large squares, it should make sense to you, okay, if I only counted 4 large squares, and Britt just told me if I can count 10 large squares, it makes it a lot easier. If you only count 4, do you agree that if you multiply however many cells you counted in 4 squares by 2.5, what did you just do?
That made it 10.
Yes, you just figured out how many would have been in 10 squares. Makes sense? Like, again, I could make it complicated and say, okay, you did all this, you did this procedure, you loaded your hemocytometer, and instead of counting 5 squares on both sides, that gives you 10, I could say you only counted 4 squares. What would your count be per microliter? Then that's when you've got to multiply it times your volume correction factor. What if I told you you only counted 5 squares on one side? Now what would you multiply that number by? 2. What if I told you you only counted 1 large square?
10.
You multiply by 10, okay? Now remember, if the problem tells you that they did a dilution, you've also got to multiply it by the dilution factor. But again, I'm trying to make it easy for you, yeah.
So let's say there was a dilution factor, and you you had 27 up there just a second ago, and the dilution was 1 to 2. So what we do is we do the 10, and then we multiply that by the dilution factor, which was 2. So we should have 54, correct?
Yep.
Thank you.
Exactly. Okay, so let's see if we understand this or not. There's a review question. I usually save this for the end, but let's go ahead and look at this review question. Let's see if we can do one of these. Number 9 on page 199. Page 199, number 9, it says, given the following information, calculate the CSF white blood cell count. Cells counted, 80. The dilution factor was a 1 to 10, and then it says the large squares that were counted was 10. Okay, now don't anybody blurt it out just yet. I want you just to think about that. All that they did different in this question is they did say that they did a dilution factor of 1 to 10. So they diluted the spinal fluid, okay? They put 9 parts diluting fluid and 1 part spinal fluid, okay? That's a 1 to 10 dilution. That's all they did different. In other words, it's got so many cells in it, they had to dilute it in order to get a count, all right? So they clearly tell you in the problem that they counted 10 large squares, all right? So somebody walk me through it, how you calculate that.
Number of cells counted.
Which was 80.
Times your number of squares.
They counted 10 squares, so there's no need to multiply by that.
Times your dilution factor.
So what would our answer be out of that multiple choice question? See? 800 is the answer. It says they counted 80 cells, they counted 10 large squares, so there's no volume correction factor. The only other thing in that question you need to worry about is they did do a 1 to 10 dilution. So 80 times 10 is 800. So the answer to that question is they counted 800 white blood cells per microliter.
Let's say that the number of squares that they counted was 5. And we take our answer, which is 800, and multiply that by 2. So we
Well, I would, okay, I think you're gonna get the right answer, but I would, if you only counted 5 squares, 5 large squares, if they still counted 80, I would write them, multiply that number times 2. 80 times 2 would be 160. If they still use this dilution factor, you take 160, multiply it times 10.
Yep, or 1600.
Yes, so it would be 1600 per microliter. Now remember, the normal white blood cell count should be 0 to 5 white blood cells per microliter. So this person obviously has something going on. There's too many white blood cells in the spinal fluid. Everybody agree with that? Anyway, manual cell counts give students fits, okay? They gave me fits. I'm sure they're probably confusing some of you guys. But just understand, I'm telling you, when you figure it out and you get it, you get it. And you'll understand, okay, all they're doing, you just gotta figure out a way to determine how many cells were in 10 large squares, okay? If they don't make it easy for you, you've gotta multiply the volume correction factor to get it to 10 squares. And you guys did this in hematology, if you'll remember those problem sheets you had to do? Where we told you like, you only count 5 squares in the center square, and remember you would have some astronomical number you had to multiply it by, like 200. Well, guess what that 200 is, all right? Or guess what that 50 is? If you only count the middle square and you count a tiny portion of the middle square, you've got to figure out, well, how many would have been in the middle square, and then you got to multiply that times 10 to figure out how many would have been in 10 large squares. Yep.
What's the normal white blood cell count?
0 to 5 per microliter. Per microliter. I think I told you guys that. I think I've got it highlighted. Just a second, I'll tell you where that's
Oh,
at.
yeah, I do.
Okay. Okay, so we know how to do a manual white blood cell count, or we at least know the basics. One thing that doctors also want is they want you to do a differential on the spinal fluid. Believe it or not, most of the time, I mean, some spinal fluids you get, they're gonna be yucky. They're gonna look like this, and you're like, oh, something bad. It's cloudy, okay? If it's cloudy, there's usually lots of cells in it, okay? But most of the time, you're dealing with a spinal fluid that's crystal clear, believe it or not. And so you have to do a manual differential. Just trust me when I tell you, if we were to take, you remember the diff sapes? If we were to take a drop of this and put it on a microscope slide and make a cut smear the way we do with blood, remember when you guys made the 3 acceptable blood smears? If you take a drop of this and put it on a microscope slide, make a smear, and then stain it with a right stain, you'll be lucky to maybe see one white cell, okay? If it's clear like this. Now, obviously, if it's bloody, all right, you'll see a lot of stuff on it. So we have to do something called cytocentrifuge technique on spinal fluid. And your book has a good picture of how to do this. I'm just gonna tell you about it, but you're welcome to read about it. This picture on page 187, I think, 187, okay? This is a cytocentrifuge technique. And I want you guys just to bear with me and tell me if you agree with this, okay? What I've got, and I'll show you guys how to put it together. I've got a little cup right here, okay? If I put, say, 5 drops of spinal fluid, that clear spinal fluid in there, and I put this in a centrifuge, it's gonna start spinning rapidly. There's a special centrifuge that spins these, okay? And that spinal fluid is gonna go through this little tube. You guys see that little tube right there? And it's basically got some filter paper between it and the microscope slide, and you let it spin for like 10 minutes. It spins at a real light spin, but it spins for 10 minutes. And they make some microscope slides, when it gets finished spinning, they make microscope slides that have a circle in the middle, okay? This circle lines up with that tube. You understand? Look, see, like the spinal fluid goes through there, the whole time it's spinning, it's concentrating that spinal fluid, and the specimen ends up being concentrated, and it's right in that circle, okay? This is called the cytocentrifuge technique. So once it gets finished spinning, you take this microscope slide and you go put it on a right stainer. And you know exactly where your white blood cells are gonna be at. They're gonna be in the middle of that circle. The few white blood cells that are in the specimen are all concentrated in that circle. How many white blood cells are you supposed to be able to count to do a proper differential?
0.
Nope, not for a diff. If you're doing a differential where you're saying, look, there's 80% segs, there's 15% monos, how many do you have to count? 100. Remember when you guys did all the differential counts in there? What made the thing go ding?
Oh yeah, 100.
When you got to 100, right? There's no way you can count 100 cells if you do a cut smear on clear spinal fluid, so you have to concentrate it. Because believe it or not, even though there's not very many white blood cells in that spinal fluid, when you do this technique, you're gonna look in the middle of that square and there's gonna be lots of white blood cells right in the middle. They're gonna be distorted. They're not gonna look exactly like they did when you guys did diffs in there, but they're still gonna be recognizable so you can count them, all right?
And we're counting different things like basophils, eosinophils, right?
The spinal fluid, the normal cells change a little bit. They're not like in blood. And we're fixing to talk all about that. So one thing I'm gonna have you do in lab next week, you're all gonna have to put one of these together, okay? And the reason is I think it's important because you'll do it at clinicals, okay? And so I want you guys to at least say, you know what, I've seen one of those before. So what it is, you have this little metal clip, just like this, okay? The first thing you do is you'll have a microscope slide. Well, that help. Anyway, it's got a circle in it. So you want to put it in here where the circle lines up with the circle, just like that. And then some of these have them and some of them don't, but this is like filter paper that's gonna take care of any excess fluid, okay? So you put it in, and then you got this little cup right here. You slide it in, and then you click it. Now, when you read about this in your book, because I know all of you are going to, it also says that you can add a couple of drops of albumin to this, and it will cause your cells to not be as distorted, okay? Because just trust me, when white blood cells go through this process, they're not gonna look like a perfect lymphocyte or a perfect seg. You can still make them out, but they're gonna be a little different. Sometimes you'll see lots of vacuoles in the cytoplasm and stuff. But it's not that they're really there, it's that that stuff happened because of this process. And like any centrifuge, you gotta have a balance. And so what I always did when I was at work, I did 2 of them. And I don't mean I put in an empty balance, I made 2 slides. You see what I mean? Because what if one of the slides breaks? You gotta have a balance anyway, so why not do it twice?
Right.
Now, you wouldn't wanna put a bloody specimen through that, because guess what would happen? If you had a specimen that was real bloody and real cloudy, and you added 3 drops of spinal fluid, when you go to look at the differential, there's gonna be so many cells that they're just gonna be all overlapping. You see what I mean? In that situation, you could just make a cut smear. It's on the clear fluids that you gotta do that process. Okay, so we're under, right-hand column on page 187, under CSF cellular constituents. It says the cells found in normal spinal fluid are primarily lymphocytes and monocytes. So how is that different than blood, guys? What's the predominant white blood cell seen in blood?
Neutrophils.
The neutrophil, right?
Yeah.
So it's telling you here in spinal fluid, the predominant cells are lymphocytes and monocytes. Okay? It says adults usually have a predominance of lymphocytes to monocytes, kind of around a 70 to 30 ratio, right? 70% lymphs to 30% monos, whereas the ratio is usually reversed in children. So if you're looking at a spinal fluid and you see an occasional mono or an occasional lymph, it doesn't mean that person has bacterial meningitis. That's totally normal. Because remember, we already established the fact that 0 to 5 white blood cells per microliter is normal, okay? So you can have an occasional one, but you shouldn't have an increase in your number. So now that's what we're fixing the lead on talking about. Skip a sentence and it says the presence of increased numbers of these normal cells has a vocabulary term and it's called pleocytosis. Everybody see that bolded word there? Okay, next column. I gotta read just about, well, I'm gonna read this whole paragraph that it's important. I'm on page 187 in the right-hand column. It says when pleocytosis involving segs, lymphs, or monos are present, the diff count is most frequently associated with its role in providing a lot of important information about the type of organism that is causing an infection in the meninges, hence meningitis. It says a high CSF white count, of which the majority of the cells are neutrophils, is considered indicative of a bacterial meningitis. And that should be easy for you guys to remember. Remember, lots of segs, it's a bacterial infection. Okay, so that's kind of similar to what you learned about the blood. But then it goes on to say a moderately elevated CSF white count with a high percentage of lymphs and monos suggests that the meningitis either comes from viral, tubercular, fungal, or parasitic origin. Now again, guys, I realize you guys are never going to make these diagnoses on people, okay? Just understand that. But for whatever reason in our field, for that registry test, they ask questions like this, okay? I'm not even saying I think it's right, because you will never pick up a phone and call a physician or a nurse and say, hey, look, I'm seeing lots of segs. You might want to be thinking about bacterial meningitis. They will tell you real quick, what are you even calling me? Okay? So I just want you guys to understand that, because the way I have to teach you guys this stuff, I'm having you figure out what's wrong with patients, basically, okay? I can't do anything about it. I gotta prepare you for the registry. But I want you guys to realize your job working down in the lab is to make sure that procedure that I just told you, that you do that as good as possible, and that you give that physician, you give him or her good information, because trust me, they know if you report out, hey, 80% of these cells are segmented neutrophils, they're gonna be the ones that tell the patient, hey, we may have something going on with bacterial meningitis here, okay? But it's real important that you do the procedure correct so you can give them information, because they only can diagnose patients based off the information you give them, okay? I mean, you know, if you call all those segs lymphocytes, that physician is going to tell them, hey, you may have viral meningitis, okay? Whereas they didn't do anything wrong, you did. You know, because they're trusting that you know what you're doing down in the lab, basically. Now, I told all you guys when you were thinking about going into this program that we didn't get a lot of recognition. You remember me? I always say that because, you know, in my eyes, you're doing the important work down there, and they just know all this stuff as far as, you know, if there's increased segs, increased, you know, limps or anything like that. But what you do is really, really important. Unfortunately, you may not get recognized for it. Okay, down in that next paragraph, I'm on 187, right-hand column now. I'm in that paragraph just above neutrophils. 3 lines above that bolded word there, neutrophils, it says cells forms differing from those found in blood include macrophages. There's some cells that I'm going to show you a picture of in a minute called choroid plexuses, epithelial cells, and spindle-shaped cells. And unfortunately, you can also see malignant cells. Okay, so I'll show you pictures of these and we'll talk briefly about them. A good chart at the top of page 188 shows you different cell types and the clinical significance of those. So you guys look at figure 9, tag 9. No, I'll tell you what, let's take a break and then we'll come back and talk about the pictures. This is a good place to stop. Be back 15 minutes.